I'm a senior data scientist in microbiome by day and a data science degree candidate by night. I'm going to talk to you today about building a deep learning classifier based off this huge cancer data set that was published back in 2020. It's from this Nature paper, very high profile, got a lot of media attention, from one of the leads in the field of microbiome, Rob Knight. So basically what they were looking for was microbial signatures that could classify multiple different cancer types. So they'd actually compiled 18,000 samples, which again is incredible in the field of microbiome, from 33 different cancer types. And the idea was that one, this would help with early cancer detection, because if you can find biomarkers in the blood that tell you about things like pancreatic cancer, which has an incredibly high mortality rate and poor prognosis, or other systemic cancers, which you might not find unless you're doing like targeted, invasive diagnostics. Further, they claim from this, and we're going to get to that in a second, that there was robust microbial signatures and that they had a large amount of contamination, but when they removed that contamination, they got down to the microbiome reads. And they looked to independently validate a model using health controls, HIV patients, and multiple different cancer types. And they looked at cell-free nucleic acids in the plasma. So that would be the perfect diagnosis area to go in at, because people get routine blood samples all the time, and you could just tack these on as like an extra diagnostic. This is actually taken from the paper, and it was adapted from an article, but this shows all the different sites that they had cancer samples from. So this was tumor, and they had a couple of blood and healthy controls, and you can see it's very prolific. It's basically systemic cancer, and they mark out different areas where this may be, promising as an aid to the field of medicine, where you have brain cancer, as a diagnostic, but say something else like skin cancer, you could have it as a prognostic. So looking at like the viral microenvironment ahead of lesion formation. So this could be like a high-risk virus, and then response to treatment. We know that bacteria metabolize a lot of drugs, a lot of xenobiotics, and they can turn them into inert forms or even toxic forms. So it's very interesting to know which bacteria they're potentially for how well the treatment is going to impact the patient. This is when we get to the controversy and why I'm interested in this, doing this study. The paper was actually retracted 3 years post-publication. It had already accrued 861 citations, which is huge, as anyone in academia knows. And what they'd found in this paper was actually there was issues with the bacteria and the bacterial abundance calculations. And actually there was a lot of human DNA getting in there. So this was giving away an RNA. So this was giving away the body site. So you were getting this classification of body site, but it was actually more the host signature than the microbial. So this was retracted, and it was agreement with the original authors too that it should. So for this project, I decided to split it into two. I was incredibly impressed with the autoencoder assignment, because I do a lot of dimensionality reduction analysis for microbiome data. So this for me kind of blew my mind, so I thought that was very cool. And then part two is a feed-forward, dense, fully connected neural network. And this would be for classification. I decided to focus on the viral microbiome fraction, because this was the fraction that they claimed to not have had issues. So I thought, I want to reduce my data set so that my laptop doesn't explode. And at the same time, I should probably look at the section that doesn't have the major overriding host signal. Just to quickly go through the data preparation, I looked at the RNA viruses. I only included cancers with over 500 samples because I wanted to be able to train my model, have test, train, validation data sets, and I didn't want to have a huge bias in the representation. This left me with 10,000 samples from 17 cancers. The first processing I did after this subsampling was actually due to microbiome data being compositional. I converted it to relative abundances. So this, to some degree, accounts for issues in difference in sequencing depth. It's not rarefaction, but it brings samples into range where someone might have had 10 million microbial DNA, RNA reads. Another might have had 1 million. The actual raw counts might reflect actually something to do with sample processing, more so than the actual the community. So relative abundances span quite a large range. So this is a percentile. So what I did is add a pseudo count and log. And then for the classifier, I had to convert the labels to integers, and then I did one hot encoding. So the autoencoder quickly, I literally took the code we used in the lecture because I was so impressed with how it worked with the wine classification. I also wanted to compare it to something that I do daily, which is I do PCOA on different distance matrices. And for this one, I picked Bray Curtis. It's a standard measure of beta diversity in the field. So I trained the autoencoder. I'm not going to go too much detail about this because this isn't the meat of it. And you can see after several epochs, we got a pretty stable MSC. Projection, so I did this in R because that's what I'm familiar with. You can see that this would be the conventional form that I'm used to looking at. And you can see here we have prostate and stomach cancer over here, and then we have this gradient all the way through to the different cancers, but you can see that there are separation between individual cancers. And the autoencoder incredibly recaptured this separation here, and actually there was more of a separation in my opinion. Bearing in mind this is taking, the PCOA is taking into account like axes of variation, and this is more looking at recapturing trends in the data. I still thought this was incredibly interesting and possibly would foreshadow accurate classification of some of these cancers. On to the deep neural network, you can see here I've set up my model in Keras. I basically wanted to incrementally build my model to kind of see and demonstrate the improvements. So I started off with a very basic model to start with. I had 311 inputs, right? So the RNA viruses. I put that through a cascade of dense connected layers with ReLU activation functions, and then obviously a softmax at the end because I want to do classification. I read this for 100 epochs, and I used the validation data with a batch size of 32. And you can see here massive overfitting to the training data and pretty poor accuracy on the validation data. So this is a great place to start from, in my opinion, because we can pull these in towards each other and get this model to be more generalizable. Second thing I did was I added some extra layers, and as you can see, it didn't make much difference. I decided to tackle this overfitting. So I came in with some dropout layers. You can see I just basically slotted them in between every single dense layer. I didn't want to explore that too much. I wanted to just see if I could pull this overfitting down fast. And you can see it worked actually pretty well. Like we're seeing over the first 20 epochs, they're comparable, and then there is some divergence later on. With the accuracy on the training data not flattening out yet. Obviously, it's not fully fitted just yet, but we do for the validation data. To tackle that even further and kind of try and reduce this overfitting a bit more, I added in L2 regularization. I didn't fine-tune this parameter. I just used a default of 0.01. All the hyperparameters from now on I will fine-tune in this presentation. And you can see that actually closes the gap pretty nicely. Over the 100 epochs, you can see that it's softening out. And even though the model's not performing fantastic, you could say that it's more generalizable. We'll go into hyperparameter tuning on the ADAM optimizer. So I did learning rate. This had a huge impact, as you can see, on accuracy. The clear winner there, 10 to the -3 on the learning rate. Then I tuned the other optimizer hyperparameters. Beta 1, you can see from these, not really having a substantial impact. So I picked the optimal again. I'm trying to eke out as much accuracy as possible, but model summarized here with the hyperparameter tuning and the model architecture, and you can see that it's not the best classifier on the planet, but the generalizability between validation and training is not terrible. Like I said in the start about body site features, could it be an indicator of, say, the microbiome microenvironment? You know, like I'm detecting stomach cancer really well because all those viruses are leaking into the blood. You know, from the stomach. It's not necessarily they're driving the stomach cancer. They're just a signature in the blood of maybe a perforation or some sort of breakdown of the barrier. And you can see here where I've plotted out the percentage of the cancer that was accurately classified and the percentage of the body site. So we see the stomach cancer classification is pretty fantastic. Dropping to ovarian, see a tiny little gap where body site is slightly, the shared body site in the classification is slightly higher than the cancer. So this means this could be cancers from the same body site. See, like anything to do with the reproductive tract could all get compounded if the viruses are the signature, more of the microenvironment than the actual disease. But you see, actually, for even the very poor classifications, it seems to be lining up. Until we get to lung, we see that an additional 10% of cancers classified, though wrongly classified, were classified to the right body site. You see this descending until we get to some very interesting ones down here, which I'm just going to look at quickly in more detail. Uterine, 0% accuracy, 0% the right body site. So that to me was very intriguing. I got a history in vaginal microbiome, so that seemed very suspicious. Rectum adenocarcinoma, 65% correct body site, 0% accuracy. Now we know the GI tract, there's a lot of different cancer types in there, so this was very interesting. Kidneys, even though they're interesting, I'm not going to focus on those two. I'm just going to quickly look on the top two. For these two cancers, what was the top misclassification? Let's put it that way. So you can see here that for rectum adenocarcinoma, colon adenocarcinoma was the highest one. Makes sense, they're very close proximity. The actual distinction between these two physical sites is actually a matter of, you know, debate. These could be right next to each other. So it makes sense that the same viral microenvironment is driving the signal. And possibly it's just a perforation of the colon, rectum, whatever, leaking into the blood or a barrier breakdown. So that's interesting. For uterine carcinosarcoma, so this is of the cells, the skin cells. The top one's prostate adenocarcinoma. These two should never meet, right? Uterine and prostate. So this was incredibly interesting to me. I suspect it's just that the uterine viral microbiome is very, very low diversity, if non-existent, and then you're getting all this bleed through of other signals. My classifier presentation and my autoencoder, I think the autoencoder did a fantastic job. We also saw that that separation kind of ended up reflecting in our neural network. Though it had poor accuracy for some body sites, it was actually pretty good. And if I was to consider further developing this, I might explore other optimizers, activation functions, architectures. I might pull in actually the bacteria, do some cleaning myself. And then finally, since the microbiome data is compositional in nature, there's other normalization methods to handle this. So I might try something like a log ratio, centered log ratio on these relative abundances, sorry, as opposed to relative abundances, or other data transformation. So I think there's a lot to be played with here for future work. So thank you for listening. That was my presentation.